scene script. Have you ever wondered how your body executes complex tasks with such precision and coordination? It's a marvel, isn't it? This awe-inspiring functionality hinges on the silent work of neurotransmitters, the body's very own chemical messengers. These messengers control everything from our heartbeat to our ability to learn and remember. Let's dive into the intricate world of neurotransmitters. The star of today's discussion is acetylcholine, a primary neurotransmitter with a critical role in the functioning of our body. This versatile chemical messenger is involved in several key processes, including muscle contractions, heartbeat regulation, memory formation, and more. The creation of acetylcholine, like other neurotransmitters, is an intricate process known as biosynthesis. This process begins with simple molecules that undergo a series of chemical reactions to transform into the neurotransmitter we're familiar with. In the case of acetylcholine, the process starts with two raw materials: acetyl-CoA, a product of cellular respiration, and choline, a nutrient we consume in our diet. A special enzyme, choline acetyltransferase, fuses these two components together, resulting in the birth of acetylcholine. But the journey doesn't end there. Once acetylcholine is synthesized, it's stored in vesicles, tiny storage units within nerve cells, until it's needed. When a nerve impulse arrives, these vesicles merge with the cell membrane, releasing acetylcholine into the synaptic cleft, the narrow gap between nerve cells. From there, acetylcholine binds to receptors on the receiving nerve cell, transmitting the message forward. The story of acetylcholine's life concludes with its swift breakdown by an enzyme called acetylcholinesterase. This crucial step prevents acetylcholine from lingering in the synaptic cleft, ensuring that nerve signals don't get stuck on repeat. The breakdown products, acetate and choline, are then recycled back into the cell, ready to begin the process anew. Understanding the biosynthesis of neurotransmitters is the first step in unraveling the mysteries of our body's communication system. So, let's continue peeling back the layers of this complex system together in our next segment. Once these neurotransmitters are synthesized, how do they reach their destination? Well, let's take a closer look at the fascinating journey of one such neurotransmitter, acetylcholine, from its genesis to its purposeful release into the synaptic cleft. Acetylcholine, like all neurotransmitters, begins its journey in the neuron's cell body, where it is synthesized from choline and acetyl coenzyme A in a reaction catalyzed by the enzyme choline acetyltransferase. Once it's formed, it's then packed into vesicles for storage and transportation. Now, here's where the release comes into play. When an action potential or nerve impulse reaches the end of the neuron, it triggers the opening of voltage-gated calcium channels. This rush of calcium into the neuron prompts these vesicles to fuse with the cell membrane and release acetylcholine into the synaptic cleft. This process is known as exocytosis. The next part of acetylcholine's journey is short-lived but vital. It diffuses across the synaptic cleft and binds to specific receptors on the postsynaptic neuron. This binding triggers a response in the receiving cell, allowing the original message to continue its journey along the neural pathway. However, this neurotransmitter's work is not yet done. To prevent overstimulation of the postsynaptic neuron and to prepare for the next nerve impulse, acetylcholine must be rapidly broken down. This is where acetylcholinesterase comes into the picture. This enzyme, located in the synaptic cleft, swiftly breaks down acetylcholine into choline and acetate, effectively terminating its action. The choline can then be taken back up into the presynaptic neuron, recycled, and used to create more acetylcholine ready for the next round of neurotransmission. So, from synthesis to release, from action to metabolism, every step in the life of acetylcholine is intricately orchestrated. This process is not only fascinating but also crucial to the smooth functioning of our nervous system. As we see, the release and metabolism of neurotransmitters are as crucial as their creation. But how does acetylcholine know where to go and what to do? It's all thanks to cholinergic receptors. These are specific protein molecules that acetylcholine binds to, acting as its guides. Now, there are two main types of cholinergic receptors we need to talk about: muscarinic and nicotinic. Muscarinic receptors are G-protein coupled receptors, and they are found in many places, including the heart, lungs, and gastrointestinal tract. They come in 5 subtypes, each with its own distinct structural features and functions. On the other hand, nicotinic receptors are ligand-gated ion channels. They're mostly found in the nervous system and at neuromuscular junctions. Nicotinic receptors have two main subtypes, one in the muscle and the other in the neurons, each with its unique structure. These specific receptors act like GPS for acetylcholine, guiding it to its destination. So it's not that acetylcholine knows where to go, it's that these receptors tell it where to be. Now, what if we could manipulate the actions of acetylcholine to treat various conditions? This is where cholinergic agonists and antagonists come into play. These agents have the fascinating ability to mirror or hinder the action of acetylcholine, creating a range of potential therapeutic applications. Let's begin with cholinergic agonists. These are substances that can mimic the action of acetylcholine. One example is carbachol. It binds to the same receptors as acetylcholine, tricking the body into thinking it's the real deal. This imitation game allows carbachol to stimulate the same responses as acetylcholine, making it useful in treating conditions like glaucoma, where increased fluid pressure in the eye can cause vision loss. By imitating acetylcholine, carbachol can help reduce this pressure, promoting better eye health. But what if we want to do the opposite? What if we want to block the action of acetylcholine? That's where cholinergic antagonists come in. These agents, like dicyclomine hydrochloride, work by binding to acetylcholine receptors. But unlike our friendly impersonator carbachol, they don't activate them. Instead, they block acetylcholine from binding and initiating a response. As a result, they can help manage conditions like irritable bowel syndrome by reducing the contractions of the smooth muscles in the gut. What's particularly intriguing about these cholinergic agents is their specificity. They can target certain types of cholinergic receptors, allowing us to fine-tune their effects and reduce unwanted side effects. For instance, some agents may target receptors in the heart, while others may target those in the lungs or other parts of the body. The manipulation of acetylcholine's action by these cholinergic agents opens up a world of possibilities in the treatment of various conditions. They provide us with a powerful tool to control the narrative of acetylcholine and its effects on our bodies. With these agents, we can control the narrative of acetylcholine for better health outcomes. This highlights the power and potential of pharmacology and how understanding the nuances of our body's systems can lead to innovative and effective treatments. But what happens when there is a need to inhibit the actions of acetylcholine completely? Well, we dive into the world of ganglionic and neuromuscular blockers. These are classes of drugs that play a crucial role in modern medicine, particularly in surgeries and critical care. Let's begin with ganglionic blockers. Simply put, these are drugs that block the transmission of nerve impulses in the autonomic ganglia. That's the place where neurons communicate with each other. They achieve this by inhibiting the action of acetylcholine, our star neurotransmitter for the day. Ganglionic blockers, such as mecamylamine and hexamethonium, work by blocking the nicotinic receptors in the ganglia. This blockade prevents acetylcholine from binding to these receptors, thus inhibiting the transmission of nerve impulses. Effectively, this puts a pause on the communication between neurons. Moving on, we have neuromuscular blockers. These come into play at the neuromuscular junction, where nerves meet muscles. Neuromuscular blockers, such as rocuronium and vecuronium, inhibit the action of acetylcholine at this junction. They do this by blocking the nicotinic receptors on the muscle end plate. Now, what's the result? These blockers prevent the muscles from receiving signals to contract. This causes muscle relaxation or paralysis, which is vital during surgical procedures to prevent involuntary muscle movements. So while our neurotransmitter acetylcholine is essential for many bodily functions, there are times when its action needs to be inhibited. That's where ganglionic and neuromuscular blockers come in. By blocking the action of acetylcholine at different points in the nervous system, these drugs allow us to control how our body responds to certain stimuli. They can either slow down or completely halt communication between neurons or the transmission of signals from nerves to muscles. And there you have it, the fascinating role of ganglionic and neuromuscular blockers in pharmacology and medicine. These blockers provide us with yet another tool to regulate the actions of acetylcholine when necessary. So we have embarked on a fascinating journey from the creation to the manipulation of acetylcholine. We've delved into the world of cholinergic agents, starting with the intricate process of neurotransmitter biosynthesis, release, and metabolism. We've seen the beauty of acetylcholine's life cycle, from its birth in the neuron to its degradation by acetylcholinesterase. We've touched on the structural features of cholinergic receptor subtypes and how these tiny gatekeepers control the flow of information in our bodies. We've also explored the role of cholinergic agonists and antagonists, the yin and yang of pharmacology, and how they can either mimic or block the effects of acetylcholine. Finally, we examined ganglionic and neuromuscular blockers, including carbachol and dicyclomine hydrochloride, and their significant roles in medicine. These substances, with their unique abilities, have the power to alter and control the way our bodies function. Remember, these tiny molecules hold the key to our body's intricate communication system. Subscribe for more because you won't find me later.